Well, good afternoon, everyone, and welcome. It's good to see everyone who's here in the room, and I know that we have a few dozen people online as well. So welcome to everyone for our collegiate lecture this afternoon. I'm Ann Curzan, Dean of the College of Literature, Science, and the Arts. And these lectures are always one of the real highlights of the year as we get to celebrate outstanding faculty and learn from them. This is our last collegiate lecture of the year, and I want to thank Julie Sparkman, who is out in the hall, and Olivia Steele, who help us organize these events, and the IT team who keep everything working so we can do this, and the event staff, since we will have food and drink afterward. And I encourage all of you who are here in person to stay and continue the conversation. For those of you who are on Zoom, we welcome you to participate in the Q&A. We will have the Q&A is being monitored. And so as we go or at the end, you're welcome to put a question into the Q&A and we will catch those as well. So before I introduce our honoree, Professor Nils Walter, I want to speak about the namesake of this professorship. So this professorship was named to honor Dr. Francis S. Collins, who was a Howard Hughes Medical Institute investigator and a gene hunter at the University of Michigan. Dr. Collins was a faculty member on campus from 1984 to 1993 and continued serving as a professor of internal medicine and human genetics until 2003. Dr. Collins led the International Human Genome Project, culminating in the completion of a finished sequence of the human DNA instruction book in 2003. He directed the National Institutes of Health National Human Genome Research Institute from 1993 to 2008 and served as director of NIH from 2009 until 2021, which, if you do the math, meant that he served under 3 different presidents. He received his PhD in physical chemistry from Yale University, where he first began studying DNA and RNA. Recognizing their significance prompted him to switch fields. He enrolled in medical school at the University of North Carolina, where he received his MD. Dr. Collins is an elected member of the National Academy of Medicine, received the Presidential Medal of Freedom in 2007, and the National Medal of Science in 2009. We can see why this person deserves to have a professorship named for him. We're delighted, such a great choice. And in 2017, he returned to U of M to present the medical school's commencement address. And now to talk about Professor Nils Walter. Professor Walter has been credited by colleagues with making, and here I quote, important contributions to our understanding of the structure, function, and dynamics of RNA. As colleagues state, and again I quote, his work has had a major influence on the field and his efforts have established considerable momentum in both single molecule microscopy and RNA biosciences at the university. Professor Walter's research is funded by leading entities such as the NIH and the National Science Foundation, and he's had an impressive number of publications to his name. Since 2017, alone, he has authored more than 50 papers and has an H-index of approximately 60. His articles can be seen in top journals, including Proceedings of the National Academy of Sciences USA, Nature Cell Biology, Journal of the American Chemical Society, and many others. Professor Walter received his PhD from the Technical University Darmstadt and the Max Planck Institute for Biophysical Chemistry. He joined our faculty in 1999 as an assistant professor of chemistry and then rose through the ranks to professor in 2009. He serves the university in many capacities. He's founding director of the Single Molecule Analysis in Real Time Center and co-directs the Center for RNA Biomedicine, for which he was instrumental in attaining its 5-year, $10.2 million funding through the Biosciences Initiative Award in 2018. He is also associate director of the University of Michigan Post-Baccalaureate Research Education Program. In 2020, he was appointed faculty director of the medical school's microscopy core in the biomedical research core facilities. Professor Walter is a popular teacher and mentor. In 2021, he received the Student's Choice Faculty Mentor Award of the Cellular and Molecular Biology Graduate Program in the medical school. He continues to receive stellar teaching evaluations for his courses and is known, and here I quote, for his tremendous service work promising to transform the diversity of our graduate recruiting efforts. Accordingly, he received in 2015 the Harold R. Johnson Diversity Service Award, which is a university level award. Among his many other honors, he is being elected a Fellow of the American Association for the Advancement of Sciences in 2011. He received the Faculty Recognition Award here at the university in 2013, the RNA Society Mid-Career Award in 2017, and the Open Science Practice Award from the Chan Zuckerberg Initiative Neurodegeneration Challenge Network in 2021. He continues to expand upon, and again I quote, an exceptionally broad and deep record in research service as well as teaching at the University of Michigan, and is recognized as one of our top faculty in research productivity and funding. These come from the external letters that we receive as part of these professorships. So we at the College of Literature, Science, and the Arts find it most fitting that Professor Walter be named the Francis S. Collins Collegiate Professor of Chemistry, Biophysics, and Biological Chemistry. Please join me in congratulating Professor Walter. It's also one of my favorite parts to embarrass my faculty colleagues by talking about them in their presence, talking about all the amazing things they have done. So Nils, if you're willing to come join me, we have a tradition now where I get to ask 2 questions before we turn things over to sort of humanize the people behind the science. So the first question is, how did you discover your field?
So to me, that came as an undergrad student back at the Technical University of Darmstadt, as we heard, where I was studying for biochemistry and read in this book about RNA enzymes or short ribozymes that at the time were relatively newly discovered. So that ages me a little bit here and reveals my age, I suppose. And we're getting the Nobel Prize a little later, and seemed like a great idea to, or a way to understand how maybe life originated on Earth, because RNA is the only biopolymer that we know that can store information in a sequence as well as fold into complex 3-dimensional structures to act as an enzyme. So it solves the chicken and egg problem of what could have been first to discover. And ultimately, myself and others who went into the field at the time always felt probably that if RNA played a big role at the beginning of life, why isn't it playing more of a role today, where, I mean, at the time when I studied, we still thought it was mostly a transcript that carried information from the genome to the ribosome to be translated into proteins. As it turns out, of course, and this was Francis Collins's work ultimately, by sequencing the human genome, that most of the genome is actually not coding for proteins and are rather coding for RNAs that ultimately then regulate what's going on in a cell and make 200 different cell types in our bodies possible. And this is, of course, what drives our work today and has led to many other Nobel Prizes in the meantime. So a very exciting field to be in.
So thank you. And then my second question, and then I'm going to get out of the way, is what you find the most rewarding part of the work. And sometimes I phrase it as what gets you out of bed every day to do the work.
I really appreciate that question also. Ultimately, so I come from Germany, of course. I've spent half my life in Germany, half my life in the US. And the German understanding and the European understanding when the first universities were founded in the 1300s was of a scholar being a teacher and researcher both. And this idea that you can actually teach what you research in a lab and bring this together, teaching both to students in your lab, your mentees, as well as to others that you have in your classes, is really very, very satisfying and brings me out of the bed every day.
So let me turn it over. Get out of the way.
So thanks, Dean Kurschen, for this introduction, for the very kind words, for the many words that describe what I'm doing, what I've been doing. I also should say that this is a great honor. It's humbling to receive an award like this. And it certainly drives home the idea that we have to work towards improving life around us, and that getting such an award is really reminding us that we have an obligation here as scholars. And so I also wanted to thank Julie and Kyle for setting things up here, and Aaron for taking photographs and all that. So there's always a support team that contributes tremendously. So thanks to all of them. So what I wanted to do today is give you a little bit of an overview of how RNA might have spawned life on Earth. I gave this already as my answer to the first question, and how it then also fuels modern personalized medicine. And all of this I will bring back to what my own research, my group's research is like. So first, to understand the future, we have to understand the past. And when the past really reaches back into the primordial world, this is what really fascinates me. My PhD was in vitro evolution, understanding how this process works. And if you think back of the primordial world that at the time had an atmosphere made up of gas molecules mostly, with lots of thunder and lightning here, discharge of energy, how did we get to this? Our current world here is a question that really has humanity has pondered for many millennia. The Stonehenge is just an example where people came together to ask their gods how this might have happened. And for a chemist, of course, the answer is, well, it's all chemistry. Right? There's chemical evolution that leads to self-organization in a way that's deterministic always, that has to happen, and then eventually biological evolution. And I will take a couple steps to kind of understand how we believe this might have happened. So if you look at this primordial world atmosphere here, it has lots of molecules that are kind of toxic today, from carbon dioxide, well known as a greenhouse gas, to ammonia, which is toxic, to hydrogen cyanide, actually, that famously the German spies always take when they are caught in the movies, right? And so ammonia also something that you don't want to breathe today, right? And I'm just reading a novel by Andy Weir where the aliens come from an atmosphere that's only 29 times our atmosphere density of ammonia. So how can we actually survive in this, right? And so a key experiment was done not far from here at the University of Chicago in 1953 by Sandy Miller and Harold Urey, which basically took all these molecules, these gas molecules that are inorganic molecules, plus water, and they boiled them in a reflux setup like this, where they would boil it here, the vapor with the gases go through a vial here that has an electric spark, basically discharge from the power supply, and then they would condense the water back. And you can do that for hours, days, weeks, and after about 2 or 3 weeks, you can take it back out and then look carefully inside. And what you find is that all the basic building blocks of life as we know it are found in this sort of experiment, from all the amino acids to the nucleobases, ATGC or uracil in the case of RNA, that make up the nucleotides that make the building blocks of information storage in us, sugars that build the backbone of nucleic acids of RNA and DNA, and even lipid molecules that, of course, form membranes that enclose these molecules and would lead to a biological evolution in the form of lipid droplets. And of course, nowadays are famous for the lipid nanoparticles that delivered the mRNA vaccines. And so in the 19th century, Ernst Haeckel thought of this as a primordial soup. And since then, of course, the thoughts have been a little refined. And ultimately, the thought is that this very dilute ocean with these molecules in it that existed in the primordial world probably wasn't enough, and you needed surfaces of minerals. I'm myself a mineral collector, perhaps because of that, that align these individual building blocks of an RNA molecule, for example, here, and align them on a surface in a way that they can polymerize together, forming random strands of RNA that have different sequences where the A, C, U, and A of the RNA are in random order. Okay? And so how do you get to biology then? And of course, you always have to fight also the fact that things decay. We all go back to dust. And so with RNA molecules at the beginning of life. Now, if you think about RNA, this is again, these are the nucleobases here in blue, the sugar, the ribose here, and then the phosphate backbone. And they can make base pairing. And that is fundamentally the ticket to get information stored and ultimately inherited by the next generation of molecules. Of course, there are no fossils that would document how the RNA world really looks like. So they are still, and maybe we will never know exactly how this all came together. And maybe there are polymers that preceded RNA. But once you have RNA, you can imagine a process by which these random strands of RNA were able occasionally to make a copy of themselves by not only by aligning building blocks that were complementary by the Watson-Crick rules, A, U, and CG going together here, and then got polymerized to a complementary strand here in purple. And if those two, maybe by heating in a deep sea vent or other ways, get separated from one another, and this red one is a catalyst to make more copies of either the complement and then copying the complement into itself, you start having molecules that can chemically evolve into more and more complex sequences and functions. And this then gives rise to the idea that an RNA world could have existed where many different copies of RNA were made, some of them catalytically active as RNA enzymes, some not. And this is a cover for my PhD thesis, actually, and so this can be captured in this idea that you go from replicating a sequence to many, many copies of it, to mutating it every so often. That creates a quasi-species, which my mentor Manfred Eigen actually termed the term. And from this quasi-species of different sequences and therefore different structures and different functions, you would get a selection on the phenotype side where ultimately the fittest survive and can replicate again and again and again. So through cycles of this, you can get more and more complex function ultimately. But then you may say, Nils, how can that work? That's not prohibited by the second law of thermodynamics, okay? Because everything goes to dust eventually. Well, as long as you have an energy source that can be an electric discharge, like in the Urey-Miller experiment, or some other form of energy may be found in form of heat or chemicals in deep sea vents, you would automatically make it possible to make more copies of this. And then you get selection in the form of only the fittest surviving that we can actually reproduce in the test tube, as I did during my PhD and many others have done. And of course, Einstein was not a chemist. He is a theoretical physicist, so he wouldn't have done this experiment. But you can imagine anyone can do this in the lab. And that has actually, but then there's also another step in here, which my chemistry colleagues will, of course, note, which is that there has to be a symmetry breaking. That is, all biopolymers have chirality to them, right? You can make an amino acid in the L or R form, S and R form, or a sugar in a D and D and R form. And likewise, not only does this exist at the molecule level here, that molecules like amino acids or so have chirality, or a virus particle has chirality, but plants clearly, or shells of mollusks or other things would have such chirality to them. So how do we get to this chirality? Well, as soon as you have molecules that have different chiralities, then there's a certain pressure that would allow some molecules to replicate better than others. And it turns out that in the end, you would actually drive at a chirally pure molecules simply by selecting those that work best to replicate and have symmetric structures that are derived from these homogeneous chiralities. Okay, so you can do symmetry breaking as rendered here by a artist. So you have two valleys, and then eventually you only continue in one valley rather than the other. And so the one type of molecule survived, but the other one that had a different chirality would not. Now, sometimes, and this goes back to an LSA theme semester, actually, on evolution a number of years back, the question that often comes up from the lay audience would be, isn't there an improbability argument? Isn't it really unlikely to happen? And the idea is basically if you had the building blocks of a bacterium that you stir up and then that reassemble, the likelihood of reassembly in the correct order just by random assembly would be very, very low. 1 in 10 to the very many numbers, high numbers. But if you're honest with yourself, then this is actually not how nature works. It's not a blind watchmaker that does this blindfolded. It doesn't know what they're doing. It just does random putting together. In fact, we are ourselves evidence that this is a very directed process, because we know that in our bodies, trillions of protein molecules fold properly every day. And this leads to Leventhal's paradox, that even a short protein, like one that has 100 amino acids, has something like 10 to the 93 different conformations that it can fold into. So how does that happen, right? Same improbability argument could apply here. But that's solved simply by the fact that a protein is not folding on a landscape like this, where this axis represents energy. It's not like a golf course where the molecule kind of goes randomly and has to find the hole there to fall into. But actually, protein in reality works forward in this kind of funneling way, where instead a protein can start anywhere on the rim, and in each case always go to this lowest energy state that we call the native state, or N here. Okay, so in other words, it's deterministic that you will always arrive here, even though you don't have to try out all the different conformations that are possible. Okay, and that's ultimately how life works. And only molecules that fold relatively efficiently will have survived evolution to this day, right? And that's why we have the proteins we have and the nucleic acids we have in our body. Another way to think about this is in fractals. So our fractal dimension as a state is higher than that of Kansas, which is flat as a pancake famously. And so the fractal dimension basically says how much diversity is there in the overall shape. Kansas, that doesn't have many hills, has a low fractal dimension. And fractals can be represented mathematically by these pretty pictures here. And you wouldn't believe it, but this is also a fractal that obviously resembles a real blue rose here. And that is created simply by a simple mathematical operation that is a complex self-similar geometric, leads to a simple self-similar geometric structure, where basically you zoom in on this particular instance here, for example, to this little arm, and that, when you zoom in, looks the same as the bigger arm. Then you zoom in on another part, deeper and deeper, and always looks the same. So it's self-similar, just like a tree has branches, and the branches branch again and branch into leaves and so forth, right? So there's self-similarity that's derived by relatively simple feedback loops that can be mathematically described and are very simple. Another argument is, again, you can do that in a test tube. And as I mentioned, people have done this. So David Patel, for example, found an RNA polymerase that can replicate itself basically, or almost can do that by in vitro evolution, by doing these cycles of mutation, of replication, mutation, and selection in the test tube. Similarly, you can make an enzyme that makes the building blocks for the RNA. And mind you, also our very own ribosomes that are found in our cells, and our most abundant enzyme on Earth, are actually RNA enzymes, probably remnants of the original RNA world. So there's lots of evidence that this is a plausible path from the origin of life to today. And that would have led then to DNA making RNA making protein, the classical central dogma of molecular biology. As you probably know, the DNA is transcribed into RNA and then translated into a protein. But as I said, Francis Collins was a mastermind together with a couple other people that showed that actually much of the human genome is not coding for protein. So if you look at the genome of the human, 98% is not coding for protein. Most of that lives as RNA, is transcribed into RNA, functioning on the RNA level. And there's clearly also a dependence of the complexity of the organism on how much is not coding in the genome for protein, with a big increase from single-celled yeast cells to fungi, for example, that have multiple cells, obviously. And to me personally, that's a little bit of a divine idea, actually. Now, you can zoom in a little bit more on RNA, and the function of RNA, of course, is a multitude of different things. These non-protein coding RNAs have functions to regulate things that go on in the cell. But then we, of course, now also appreciate the fact that an RNA virus, the SARS-CoV-2 virus, was fought by mRNA vaccines that were delivered by a vaccine into a cell and then translated by the ribosome into a spike protein that instructed the immune system to target the virus and suppress the infection when the virus had entered the body. Now, the question is, can we redirect this kind of discovery into other directions, for example, to fight cancer? And in cancer, that can work because mutations lead to cancer cells oftentimes producing what are called neoantigens, so proteins that are unique to that cancer cell are not found in normal cells. And now you can tell the immune system to please find cells that have such neoantigens on their surface and then attack them and kill them. So cancer fundamentally is a lack of the immune system to actually get rid of this particular cell. Normally, 100 billion cells in our bodies are destroyed every day, right? But one slipped by, became a tumor, makes metastasis, but we can re-instruct the human genome to actually target it and destroy it. And these yeah, and this is what I said. And of course, that has already happened. So end of last year, Moderna and Merck teamed up in a clinical trial that then ended because of compassionate use for the placebo group. That is, I mean, the people in the patient group that were treated with the mRNA vaccine against cancer, in this case, skin cancer, together with another medicine, fared much better than the placebo group that didn't get the mRNA, suggesting that really cancer is one of the next frontiers of using RNAs to our purposes. And this goes back to the idea that we need to support foundational, fundamental science. I think calling it basic research in English is the wrong word. It doesn't reflect what it actually is, right? In German, we call it fundamental Wissenschaft, so fundamental science, because it lays the foundation for everything that comes later. And that is actually represented here at the University of Michigan because Michigan has strong mRNA connections. For example, Marshall Nierenberg got his PhD in the Department of Biological Chemistry here in the medical school, and later went on to co-discover how codons in mRNAs code for protein amino acids, right? How the translation process actually works, and shared the Nobel Prize for this discovery. Åke Uhlendijk was an undergrad in our department, chemistry department, a number of years back, became, and his father actually a famous theoretical physicist professor in physics here. And Åke not only grew up here, but years later would describe the very technique that's now used to make mRNA vaccines by basically doing transcription in a test tube. Okay? And I don't think he has IP, intellectual property on it, so he's not a rich person, okay? But it all goes back to a seminal paper in the 1980s, all that's being done by Pfizer and Moderna nowadays. And of course, Melissa Moore, we had the honor to give an honorary Doctor of Science degree this past December. She is the now emeritus Chief Scientific Officer of Moderna, who was really behind formulating and pushing forward mRNA vaccines that have saved millions of lives, for which she received here from Ron Weiser as one of the regents, the honorary degree, and was with it. And just to give a preview of the kinds of things that I'm involved in, Anne mentioned, Dean Kurschen mentioned at the beginning that I'm doing things beyond just teaching and doing research. So here in the Center for RNA Biomedicine that got this Bioscience Initiative Award that Dean Kurschen mentioned, we are embarking on developing an mRNA therapeutics initiative, which would use bioinformatics to find targets, do RNA manufacturing to make molecules, medicines of different kinds, to then deliver them to a patient. And the fundamental new thing about making mRNAs or RNAs generally as therapeutics is they are information materials, just like we discussed. And as information material, you can design them in the computer. You don't have to do a 10-year trial to find a drug. You can make it in the computer, and that takes 45 days in the case of mRNA vaccines against SARS-CoV-2, for example, and many, many other things. I mentioned cancer and other things this could apply to. So hopefully we can raise some funding to do this. But you may ask, okay, what does that have to do with Nils' research, right? And so to us, to my group, really, the cell is a vast envelope to understand biology. And so if you know the cells in our bodies, eukaryotic cells, mammalian cells, have a nucleus and they have a cytoplasm, and there are many, many different types of RNAs in there. Not only the messenger RNAs that are transcribed and processed to then be exported through this nuclear pore complex from the nucleus to the cytoplasm, but they also get regulated by a plethora of different RNAs that are non-coding, non-protein coding, as I mentioned before, and that determine whether a cell becomes a heart cell, a neuron in the brain, or a skin cell, for example. And so as chemists, we want to understand these processes from first principles, from chemical principles. For example, how these machineries, for example, the RNA-induced silencing complex, which regulates gene expression of these messenger RNAs in our cells, recognize their target. How do they bind to one another? And ideally, we want to understand this from single hydrogen bonds forming, right? Which is the key of recognizing via Watson-Crick base pairing a target given a certain guide RNA that guides the machinery to regulate it. And so the tools we use in the lab are tools from single molecule microscopy. So we have a couple of different fancy microscopes in the lab that ultimately all take movies of RNA on a surface here, where we illuminate them, for example, in this setup here, through a prism, a quartz prism that's connected to a quartz slide here. And then we observe molecules that are on the underside of this quartz slide and in contact with a buffer solution here through the bottom cover slip onto a camera that's attached to an objective of a microscope. And that allows us to record single molecules because we produce what is called an evanescent field, a very, very thin light field that comes from light behaving both as a particle that bounces off when it comes in at an angle, but also as a wave producing the standing wave and quantum mechanical process at the surface where the RNA molecules are mobilized. And that gives us the low enough background to see single molecules. And if we label them with two different colors, we see them changing between colors as they undergo their function. We can also do that in cells directly when we with a slightly different setup that doesn't use a prism, but an objective here. And this total internal reflection fluorescence that works with the objective directly bringing in the green laser light here, creating this evanescent field on a cover slip, and then detecting molecules through that same objective. So those are the type of tools we use in the lab. And sometimes I still get this question of, okay, why bother using complicated microscopes to do your work? Of course, being at Michigan, I have to make a reference to football. If you think about the Michigan Stadium, right, 114,000 or so of the best friends sitting there on a Saturday afternoon waiting for the game, you will also see that if you look at it this way, you can't make out much. You can tell that a lot of Michigan fans are clad in maize. The Ohio State fans in red are only in the upper ranks. They can't get close to the field, right? But you cannot make out details unless you zoom in and recognize that somebody is very excited or very depressed, depending on how the game goes. Or ideally, you have a movie that tells you that this person gets into a very excited state, that the neighboring person doesn't quite understand why, if you look at the person then turning around. Or in this case, this person seems much more subdued than the fans around him, right? And so clearly there are differences in behavior of people, and that's the same for single molecules inside the cell. If you think of this as the cell content with all these different particles, people or molecules in them, they all go about their own business. They don't know of each other necessarily, but they interact with one another every so often and then eventually bring life to be as we know it. Now, one example of our work was done by Julia Widom, a postdoc, who's now at the University of Oregon with her own lab, and a former undergrad student. She mentored Victoria Ray, who is now a grad student at Yale. And so they worked on this, what we call a riboswitch. So riboswitch is an RNA that folds into complex structures, which we call a pseudoknot here. And a pseudoknot simply means you have Watson-Crick base pairing here in this paired region 1, and then a loop. And then the RNA folds back on itself, makes a second set of base pairing here. And then as it's being transcribed, by the RNA polymerase here from a DNA template, a certain ligand, a metabolite of the cell can bind or not. And if it does bind, that leads to complete folding of this pseudoknot here. And that terminates the RNA polymerase. That is, the RNA polymerase folds off and doesn't make the rest of the RNA. And so gene regulation has happened. This RNA does not even get made and therefore not translated into a protein that would have made more of this ligand, this metabolite, right? It's a simple negative feedback loop. If you've made enough of this guy, then you don't have to make more. You don't have to make the enzymes that would make more of it. So termination is important. Right? And ultimately, Julia and Victoria actually set out to do a single molecule fluorescence experiment where they made the RNA with two fluorophores, we call them donor and acceptor fluorophore, attached, and then immobilized them on a surface in different ways, either directly or with the RNA polymerase present, and then asked the question, how does it fold? And there are differences depending on whether or not the polymerase is there that tell us something about how the polymerase and the RNA, the riboswitch, work together to bring about gene expression regulation. And then they also ran gels. So gel is for the non-biochemist, right? Basically the content of a diaper taken into a slab and then running molecules through it at a certain speed that depends on the size of the molecule. And so this is a transcription, that reaction that is stopped at different times here. And you see that there are certain bands appearing, which have to do with this having a length of 46 nucleotides, so this one having 70 nucleotides. And what Julia discovered is that this particular band here that appears and disappears over time in the absence of the ligand, PreQ1 it's called, of that binds to the riboswitch, is much less present when the PreQ1 is added at high concentration. And so now you see this band less, you can actually quantify this, and you see that while it lingers around for a long time in the absence of this metabolite ligand to the riboswitch, in the case where it's present, then it decays much faster, it disappears over time much, much faster, threefold faster. And that tells us something about the action here, which led us to a model where the RNA can fold or unfold, but it just has been transcribed by the polymerase, sitting outside what is called the exit channel of this polymerase that has transcribed it. And then it interacts with the polymerase and it pauses the polymerase. It presses a pause button. And when the PreQ1, this ligand, comes in and binds to the RNA motif, that basically provides a kick in the butt of the polymerase that then moves it out and ultimately leads to termination. And ultimately doesn't allow the protein to be made. And so this is kind of akin to the wide receiver getting the ball, and the ball is the ball molecule ligand, and then running, right? And running out of this pause, right? And so it's also a story of David against Goliath, right? The small ligand or RNA influenced the large polymerase that is about 30 times or more the size. And more recently, in a large collaboration with Melanie Ohi's lab in the Life Science Institute and Aaron Frank, a former colleague in biophysics and chemistry, we actually obtained what is called a cryo-electron microscopy structure, a cryo-EM structure that gives us some information on this complex. And this, you can see, is the Pac-Man kind of structure of the RNA polymerase here. And this is where the RNA comes out of this exit channel that I mentioned. Here, this is just a different view, 90-degree turned. And what we discovered is basically that the two conformations of the RNA in the absence and the presence of this ligand that it binds is quite different. And you can see that the RNA outside the exit channel of the polymerase here in the background is twisting. And that ultimately, when it binds the ligand, extends the RNA end into the active site in a way that it now can be extended. So elongation, that is, I mean, the RNA is further transcribed, right, as this happens, as the ligand binds. So getting a picture of that is important. And we also discovered that there's actually a conformational change in the entire RNA polymerase. So again, David against Goliath, the small ligand can trigger the RNA to twist to the exit channel. And that ultimately pushes on the polymerase, the domains around the exit channel in a way that it unswivels, as it's called. So it clamps down on the template and starts moving, transcribing more. And you may ask, what practical application does this have? Well, ultimately, we can learn how to inhibit bacteria from growing, right? Antibacterials, antibiotics, there are not enough of them in 2050. It's expected by the WHO, World Health Organization, that will be drug-resistant bacteria will be a huge problem. There are many emerging strains already. And so by learning how the machinery works, doing this fundamental research, right, we hope to ultimately be able to develop new drug approaches, new medicines against bacteria. And briefly, I want to also bring up a second example here. This is developed by two graduate students, a chemistry grad student, Situ, and cellular molecular biology graduate student, Amia, in my lab, a tool that we call iSherlock. So we are little Sherlock Holmeses, as you might have noticed, in the lab, where we use our objective type total internal reflection, or TIRF microscope that I mentioned earlier, bring in a laser beam here in red, but we bring it in not to completely bounce back, but actually to be deflected like this. And then that illumines all cells that we have in a dish here that grow. These are human cells called HeLa cells. And then we can microinject these cells that are alive with a microRNA, one of these regulatory guide RNAs that I mentioned at the very beginning, with a fluorophore label on it, with a dye molecule that we can detect again. And then we can do particle tracking. We can track the molecules as they diffuse through the cell. This is particle tracking at what is called super resolution. Nowadays, these microscopes can break the diffraction limit. Or we can measure stoichiometry if we fix everything in space. So we kill the cell effectively, and then we can measure how many molecules have come together into a single complex. And so this is how this works. You see actually the microinjector needle parked over these cells, and every so often touching down and injecting a little bit of volume with the fluorophore label, this labeled RNA molecule. And then we can do imaging. If we do it the wrong way, then we can't see much. But if we do it the right way with this near total internal reflection or high-low microscopy, then we see individual particles diffusing in the cell, and we can track them, we can follow them in the cell, and we can see individual ones behaving in different ways, just like people in a stadium, where they diffuse either in Brownian motion or they move along a track, and they are transported probably with the messenger RNA that they're regulating here. And we can learn all sorts of things by observing single molecules, single particles in the cell. In fact, we clarified some confusion in the field that a microRNA, this is this regulatory RNA that's bound by this RNA-induced silencing complex or RISC complex, can regulate a messenger RNA. We can observe the diffusion only once it's bound to a messenger RNA, because otherwise it blurs out. And then we can, after about an hour, tell that it's probably the messenger RNA is probably inhibited in translation, so it can no longer make a protein. But it takes another 7-8 hours, and we have data to support this, for the mRNA to actually be degraded. And this clarified a confusion in the field where people had different observations, thought, oh, it's just translation is inhibited, and other people thought it's only degradation. It's actually both, but one after the other. Again, chemistry helps by physical chemistry, by time-lapse experiments, by timed experiments, to really measure the kinetics and figure out, okay, something can go fast, and then something happens later in the slower time scale. And we can measure that directly and ultimately see the elephant for what it is. It's not a spear, it's not a snake, it's not a wall or a rope or fan, as blindfolded men might see it, but actually we can see it in its entirety by using the tools that I described. And application here might be to redirect microRNAs to fight diseases, right? These regulatory RNAs, to utilize them. Again, from understanding the principles, we can ultimately utilize them. And so that ultimately gives this picture that molecules in a cell each go by their own path, right? Everyone does their own little thing, takes a few steps, but the whole is greater than the sum. As you see here with the Ohio State marching band eating a Michigan player, right? Because they can do that, even though every single one takes only a few steps, together they form a Tyrannosaurus Rex and eat the player, right? And so it's synergy from all of them, all the parts working together. So in summary, I feel, and I hope I mean, if you didn't get much of the talk here, the take-home message is really that RNA connects us to life's chemical origins, right? And this is important to us chemists, that in the future, RNA may save many more lives. So hopefully, we can help with our mRNA therapeutics initiative. But this will come. It's $1 billion industry expected to help all sorts of diseases from individuals that have a disease because of particularly genetic mutation that can be helped by design of a new drug on the computer and giving it compassionately, to cancer, which affects so many people. And ultimately, RNA can be understood using these single molecule fluorescence microscopes that we have in the lab. And with that, I really need to thank all the people in my group that play Sherlock Holmes. A lot of collaborators, some of their work I mentioned already, funding, NIH, NSF, but also Jane Zuckerberg Initiative, and really science depends on a diverse team and collaborators, right? Diversity matters at all levels, including in research and teaching. So with that, thank you very much for all your attention.
No, that was terrific. Thank you. And you've left us time for questions. So I think, please call me Anne, but I'm just going to take questions from the floor. And I know we may have some coming in online.
All right. Any questions? Everyone's so overwhelmed. Ted, thank you.
Great talk. I enjoyed it. So in your work, you also described that some of the folks in RNA are doing synthetic modifications, where you kind of you work on the natural kind of thing, right? And I guess in terms of maybe mixing the two together, have you started to move in that direction? I know that's for drug discovery. That's like the thing, and you're sitting next to a person in the front row who does this for a living, I think, right? And so, but merging those two together, I think would be very powerful, right?
It is. So, I mean, for one, I have a starting collaboration with Pfizer, who are very interested in quality control of the mRNAs. And those are, of course, modified with one methylsuryouracil, so a chemical modification that's very important for the stability and not for not raising the inside the cell some immune response-like responses that kills the cell and doesn't let it overexpress the messenger RNA. And so clearly there's an interest of industry, for example, to do the molecular diagnostics, in this case, to understand how diverse or uniform their preparations of mRNA vaccines is, depending on the modifications they put in and so forth. But generally speaking, the RNA molecules we use can just as well be modified. We put fluorophores on them, that's a chemical modification. But there are many other modifications that could be used. And one of the things that I've been interested in for a long time is to utilize these smaller RNAs, these guide RNAs that are used by this RISC complex that I mentioned, or the so-called CRISPR genome editing technology uses oftentimes chemically modified RNAs. What is less well understood is how they are taken up by the cell when they're delivered to a cell with some sort of a carrier, and how quickly they are taken up, where they go. And about 10 years ago, I was about to start a collaboration with Novartis on this very question of how these small RNAs actually get delivered when they have all these chemical modifications. And then they closed shop in Cambridge and threw out 35 of their researchers. So it depends on an opportunity presenting itself. But yes, I agree. Very important. Yeah, excellent.
Thank you, Nils, for the very nice talk. I am now conveying a question from Jonathan Wynn online. The question is, do you know why the RNA vaccine has efficacy in melanoma? What about solid tumors?
Right, so to be determined, right? So melanoma has a much lower barrier for delivery, right? Because it's a skin cancer, you can deliver it directly to the skin. So anything that's on the skin or in the liver, we can deliver RNAs to. Liver, because if you inject an mRNA into the bloodstream, that's where it ends up, right? because delivery vehicles basically in the blood are filtered through the liver, maybe through the kidneys, right? And so the two organs basically accumulate them because that's their biological function. It's much harder to deliver to specific tissues or to a tumor specifically, which is, I think, still the hurdle. This RNA delivery that I mentioned briefly is really the key to really unlocking all the possibilities that RNA has. On the other hand, there's hope. There was a Nature paper just a week or two ago, Fei Zhang from MIT, who developed a bacterium into the delivery vehicle, which is a bacterium that naturally makes a little straw basically on its surface to inject proteins into other cells, and you can redirect it to any cell by specific recognition mechanisms. And while they only used it in this Nature paper to deliver proteins, there's clear indication that it could be used for RNA with some more engineering, right? There are many other techniques. I mean, there's thousands of groups, many companies that work on delivery problem, which then would allow addressing solid tumors in the body by recognizing them, perhaps also through their neoantigens on the surface, right? And delivering directly to them. Excellent question.
Thanks, Nils, for a great lecture. Yeah, it's really fascinating to think about the origin of life and RNA playing a big role, and you can see the RNA molecules competing, and whoever is the most suitable for the environment and can replicate will win, and you keep going, and you have cells, and then you have steps in evolution where you know, these are big steps where it's a little unclear how they take place, and one thing that has to do with RNA is the insertion of introns that then needs to be removed. So that step in evolution, when you started getting an intron in, that you already needed the mechanism to take it out, otherwise it would be chaos. So any thoughts about how those steps in evolution may take place?
Yeah, and one fascinating thing to me is that our genome today is, of course, a historic recording of what happened to our ancestors and in the primordial world billions of years ago, right? It's a recording over this entire time, right? So what we have now in our genomes might have been installed like introns a billion years ago, maybe, well, not that long, but maybe 100 million or 200 million years ago. And they probably derive from what are called transposons, so retrotransposons, which are unique entities that can replicate just like a virus. HIV, in a way, acts like one that can make more copies of itself, leave the host, infect a new person, and ultimately bury itself in the genome. There are HIV copies in the genome of people that have been infected, even if they don't have acute HIV infection now. And those are very difficult to get rid of, and that's why, I mean, drugs have to be given lifelong. So clearly there's evidence that this happens, right? You get parasitic genetic elements to insert themselves into the genome, and the first ones probably contained a machinery to cut themselves back out to replicate by way of the host cell machinery, and then make new particles that could infect other cells, so they spread. And this machinery probably was co-opted by the cell, presumably because there was some evolutionary benefit to keeping some of those introns around. We know that they can help a cell diversify its genome. There's evidence that plants, for example, under high stress, make a lot of transposons that then escape and find new places. And that might be part of how a plant cell that can't evade an invader as easily might get some of its genetic information going elsewhere. So there's clear evidence that there's, to this day, a lot of exchange of genetic information between cells in a multicellular organism, but also horizontally between different organisms. And many of these introns probably, or intron-like elements, drive these sorts of things. And so again, it's likely that this is an imprint of something that happened, I mean, hundreds of millions of years ago, perhaps, and got kept in the genome because it ended up having some useful function under certain conditions and helped us survive or helped the organism survive.
So I have another question from online. This one comes from Annalisa DeFeo. How can we get the field more interested in microRNA therapeutics? There seems to always be a concern with microRNA mimic integument delivery.
Right. So this is a colleague from medical school who works on this. So good question. Excellent question. Right. So the way we envision it within this idea of mRNA therapeutics is that different diseases will be best fought with different modalities of RNA. That includes microRNAs or related small interfering RNAs that use this RISC machinery. And so the thought is, I mean, there are certain siRNAs at least that are already being used in preclinical trials in animals and so forth, and possibly, I think there's even 1 or 2 drugs maybe that are in the works. To utilize these siRNAs to regulation. And so microRNAs could serve as that. There's also the idea that you can counter the effects of microRNAs in the cell. For example, cancer cells express a different set of microRNAs. And so basically countering them by a complementary strand that keeps them from regulating might help in certain cancer cases. So ultimately, I think it will be all of the above that will be used. There's this question of, at what time can we actually edit the genome to get rid of all the problems? Well, that's a little bit further away. If that worked ever, then we could potentially do a lot of things. On the other hand, it also has ethical considerations to keep in mind. And right now, you can't change all the cells in their genome in a body, only a fraction of them, and can you hit the right ones and so forth. So again, there are multiple different modalities that probably going forward will all be used for certain diseases, and some will be better for certain diseases and others will be better for other diseases, right? So. And for example, mRNAs are just very popular with cancer now because cancer is a lack of the immune system to attack that cell that somehow invades it. And so reteaching the immune system to attack that cell that escaped immune responses so far is a way to overcome that problem, right? So again, it depends on a particular disease. One of the students? Okay. No. Sorry.
Okay, and so for the final question, this comes from Fabrice Leclerc. Some small molecules acting on alternative splicing and the generation of neoantigens have been used to increase the efficacy of treatments for immunoresistant patients. Do you consider doing that, by which it seems they mean combining both RNA and small molecule drugs?
Yeah, that's an excellent question. So as I alluded to, it's actually much harder to make a new small molecule drug. And we know that because one of my students is trying that, and we are also trying to make an information material RNA drug, and it seems much easier to do the RNA drug than a high throughput drug screening, at least here at the University of Michigan. And so, so it's not trivial to execute a high throughput drug screening, which is typically the approach to find a new drug that binds an RNA and has a certain effect on cells. But if a drug is already known, then of course combinations of drugs will oftentimes be the right solution, just as the Moderna Merck trial was, because that was a small molecule drug combined, an antibody drug combined with an mRNA drug. So again, 2 different biologics, as they're called, right, being used. But generally speaking, there can be different modalities that can be combined for the best effects. And I think that will be the case, certainly. Mm-hmm.
So thank you so much for this talk, which I think was really just a beautiful and powerful example of why foundational science is so important. And as a linguist, I'm going to now use foundational science and make sure that we are using that term because I think it's a beautiful term. And I will say, having never seen you teach before, I was not expecting as much football as I saw today. But we have a little memento for you to commemorate this event. And can you all join me in once again thanking Nils Walzer for this talk?
Oh, thank you very much. Thanks.
And now we hope you'll join us for more conversation and food and drink out in the lobby.